Imagine you want to build a nice and beautiful house. You actively choose what your bedroom look like, kitchen appearance, and lobby aesthetics. Likewise, if parents can select their yet-to-born baby's IQ, height, skin color, or eye color, and many more as a customized baby, how miraculous it is! The science of biotechnology and genetics is opening a brave new world of future humans. Have you ever thought, how come people from same human race have different heights, different face structures, and diverse abilities and IQs? Although environmental factors are a reason, the fundamental cause is DNA, or popularly we call it genes. A baby acquires DNA code from both the parents. That is why we can observe striking similarity between parent and child traits. Human body is composed of billions of cells. Cell is the basic building block of human body. At the center of the cell is nucleus. The whole DNA code is stored here. What is DNA code? It is a long sequence of chemical blocks like adenine A, thymine T, cytosine C, guanine G. These A, T, C, G blocks are formed in sequence of groups like ACG, TCC, and AGG, etc. Billions of these groups is called DNA code. If DNA code is a textbook, a sentence of words are genes. Each gene is responsible for different traits of human. Let us say eye color. OCA2 gene of DNA is deciding your eye, either blue or brown or black. MCIR gene is deciding your hair, either black or brown. Your cell interprets these gene code and develop hair color accordingly. These genes are passing to next generations from the time immemorial. If any small change occurred in code sequence of gene, say from TAG to TAA, gene becomes mutated gene. It will be reflected as change in your hair color or hair texture, etc. Sometimes gene mutation is so serious that it becomes a genetic disease like thalassemia or Huntington's disease. Thalassemia is a blood-related disease wherein hemoglobin production is reduced. As oxygen carrying ability of blood to other body parts is reduced, patients feel fatigue, frequent infections, and severe weakness. Huntington disease. It is a brain-related genetic disease wherein after a certain age, human cells get killed automatically, causing related body parts become dysfunctional, memory loss and paralysis will fall upon. Until genetics has emerged, these diseases are accepted as fate in silent years, thanks to the biotechnology called CRISPR-Cas9. With CRISPR technology, we will locate the mutated part of DNA code and cut the modified code and insert normal code sequence. As in the case of thalassemia, hemoglobin is produced in bone marrow. A stem cell is collected from bone marrow of patient. Inside laboratory, mutated gene code of stem cell is edited using CRISPR-Cas9 technology. A normal gene code is inserted inside DNA code. This edited stem cell is reinfused into patient by removing older cells at bone marrow. This new stem cell generates billions of new cells, and in turn, normal hemoglobin levels are produced, solving thalassemia disease. Likewise, many of genetic diseases like cystic fibrosis, type 2 diabetes risk, or certain cancer risks are mitigated using CRISPR-Cas9 technology. Alright? Until now, the story is all about gene editing in grown-up human. Let us explore the advanced world of DNA editing in human embryo. When the first cell is formed in mother's womb, it contains the full DNA code. A few cells are separated and studied under laboratory for any mutations in genes. Such mutations are corrected by CRISPR-Cas9. This gives rise to the DNA babies or genetically modified babies. The only known such an experiment is conducted by a brilliant Chinese scientist named Jiankui in 2018. The goal of scientists is, when father is HIV positive and mother is HIV negative, delivering the baby without any possibility of HIV to the baby. He got successfully modified the gene related to HIV, and first DNA babies Lulu and Nana are born in 2018. Although intention is lofty, these unethical experiments with careless research have landed him in jail for 3 years. There is a great debate going on in scientific community towards this embryo gene editing, as in one hand it can create healthy and perfect babies with highest IQ, great strength and great appearance. In another hand, it can create inequality, as only rich can get benefited, where poor people and their offsprings will become second-class humans. Apart from ethical questions, let us explore the challenges. Most of traits are dependent on multiple genes. Say height, it depends on 12,000 genes, and human IQ, it depends on 20,000 genes. To improve IQ significantly, all these 20,000 genes need to be edited. But present CRISPR technology can make maximum 10 gene edits reliably. Next challenge is, if any accidental edits on other genes happens, it raises to severe diseases like cancers. To get genetically designed babies, science need to get far advanced, and society at large need to come to consensus on its usefulness. Until then, let us hope for the best of science.